like to introduce our eminent international speakers for the next talk. Rashmi Prasad ma'am, she is a doctorate degree and PhD in molecular genetics from Medical Faculty, German Cancer Research Center and Heidelberg University, Germany. She has a junior group leader. She is a junior group leader and associate professor at University in Sweden. Her main research interests are fetal programming and parent of origin effects in type 2 diabetes, gestational diabetes, and islet biologies. Her mentors include person like Kari Hemkin sir, Akhilesh Pandey sir, and Live Group. She has authored more than 50 publications in high impact peer-reviewed international journals such as Nature Genetics, Cell Metabolism, Nature Communications, Blood, and Lancet like Diabetes and Endocrinology. Her research is funded by her own grants with Swedish Research Councils, Heart Foundations, Lung Foundation, Diabetes Fondant. Then she is also very much known for her eminent ability of giving international lectures. Now it is a high time that we should listen and follow her advices about evolving. So ma'am, the stage is yours. You can begin with your topic.
Thank you very much for that introduction. I'm just going to start by sharing this slide. So does this look okay? Can you see the presenter view? Yeah?
Yeah, ma'am. Yeah, ma'am.
So good afternoon, everyone, and thank you very much for that introduction again. And I would like to begin by thanking the organizers and especially Dr. Banshi Sabu for inviting me to speak at this event today. And in the next 20 minutes, I will talk about fetal programming and how this can alter the risk of diabetes and a little bit about the data that we have generated in this topic. So it's very well known that a family history of diabetes is a huge risk factor. So if you have a first degree relative with type 2 diabetes, the risk of developing diabetes has been found to be approximately 40%. And what's also been reported quite often is that this risk is higher if the mother has diabetes rather than the father. And this is the reverse in the case of type 1 diabetes, where if the father has type 1, then the child has more propensity to develop diabetes. So what is being proposed and also observed to some extent is that the offspring who are type 2 diabetic have a higher propensity to inherit these risk alleles for type 2 diabetes from the mother. So this is referred to usually as a parent of origin effect. And this means that the trait effect depends on which allele the parent, which parent the allele is inherited from. And this basically links back to the DOHAD hypothesis, which is the developmental origins of health and disease, which was proposed by David Barker, wherein early life exposures can cause adverse consequences to the offspring. And fetal programming is linked with both genomic imprinting and parent of origin effects because both are determined by resource provision and restriction in early development. And according to the Barker hypothesis, the maternal exposures, both pre and post-conception, can alter fetal programming during development in utero, and they can cause adverse consequences in later life. And a lot of these include type 2 diabetes, cardiovascular disorders, obesity, and many, many more. What's also been recently reported is that the paternal exposures like smoking and obesity can also have an impact on the offspring via altered epigenetic coding in the sperm. So it's been said that the sperm contains a memory of all of the man's experiences, ranging from his nutritional status to his exposure to toxic chemicals. So what happens on a physiological level? On a physiological level, adverse exposures like diet, hypoxia, stress, like glucocorticoid exposure, then there's placental insufficiency and efficacy, and other conditions can adversely affect developmental programming during the perinatal period. And the effect is multifold. So it starts with adipose tissues where there could be excess lipid storage and high relative white fat and increased abdominal adiposity. Then there's low muscle mass. There is also impaired glucose handling due to, for example, in the pancreas, there could be reduced pancreatic beta cell mass development. And then there's immense vascular dysfunction, which could lead to cardiovascular dysregulation of the cardiovascular system and renal insufficiency due to reduced blood flow and diminished nephrogenesis. How does this programming take place at the molecular level? Well, this is a segment of a chromosome, and this is unraveled to reveal a double-stranded DNA. And this DNA is wound around these proteins called as histones. And what happens is there's an additional layer of programming on top of this through epigenetics. And for example, this could be in the form of DNA methylation of the cytosine molecules in the DNA. And then there are also histone modifications that include acetylation, methylation, phosphorylation, etc. And both of these together in tandem become a code by itself. And this will then reveal or provide information as to which gene, when, and how much of this gene should be expressed. And then what are the mechanisms underlying this epigenetic programming? So DNA methylation and histone modifications, of course, act in tandem. And they can alter the epigenetic coding of the maternal and the paternal alleles in the offspring, and this can lead to changes in gene expression. So what happens here is that in early development, there is a complete erasure of the parental specific imprints in the germ cells. And then this is completely reestablished. And after fertilization, this imprint marks are sort of maintained throughout by DNA methylases. And as the cell propagates, except in the germ cells where they are erased and reestablished for the next generation, this continues to be maintained. And so any exposures that you have during either this phase or this phase can actually alter the programming that is supposed to happen. And then this can actually alter the consequences in later life. This differing optimal fetal program trajectories of paternal versus maternal genes are predicted to represent major determinants of advantageous and deleterious programming effects, especially those that are involved in insulin secretion, insulin resistance, visceral obesity, and other manifestations of the metabolic syndrome. So this is something that's come out from a publication in 2020, literally, where it shows how the maternal and the paternal effects can manifest. So at placentation, there could be reduced placental growth. And at birth, this is manifested as low birth weight, lower muscle relative to fat, lower lean mass overall, lower beta cell mass. At infancy, there could be higher brown adipose tissue thermogenesis, lower appetite, reduced sucking, less early catch-up growth. And in childhood or juvenile or adolescent stage, this could also be less interest in complementary foods and hyperphagia for cell-rich food. And there's also earlier puberty. So this is also an impact. And all of these actually confer a selective advantage. So in the thrifty genotype hypothesis, when it was feast or famine in the early days of either hunter-gatherer situation, this would actually confer an evolutionary advantage because this would provide a survival benefit by storing all the fat against starvation. And also early reproduction would be advantageous as well. However, in the day of today, when there is more westernization, this leads to more of a thrifty phenotype hypothesis, and this confers more of a disadvantage because this leads to more insulin resistance, diabetes, and obesity. So an additional dimension is, of course, the paternal contribution, which adds to the risk. And how this happens has been it has been manifested as lower rates of brown fat thermogenesis, and also paternal programming sort of catches up much later on, and then there's a rapid catch-up effect that happens after intrauterine growth restriction from environmental causes. So there are two concepts that have been proposed for the role of paternal and maternal contributions to the fetus. So one of these is the conflict hypothesis, which stipulates that there's a genomic tug of war between the paternal programming and the maternal programming. So the maternal tries to conserve her resources so that she can increase her evolutionary fitness and pass it on to more of the offspring, potential offspring, whereas the paternal programming tries to maximize the maternal resources to this one offspring to increase the survival of the offspring. So the analogy that is most commonly used, and it's sort of one of my favorite analogies, is that the embryo development is like a car on the highway of growth and development, where the paternal programming tries to speed up the car on the highway and the maternal tries to slow it down. And the paternal would, of course, eventually catch up later on. So in contrast, another theory has been proposed, which is the coadaptation theory. And this states that there are paternal and maternal genes that have co-evolved to optimize the care of the offspring. One of the most adverse consequences of fetal programming is how this can actually have transgenerational effects. The adverse exposures not only impact the first generation, which is the F1, the developing fetus, but it can also affect the F2 or the subsequent generations by introducing changes or epigenetic alterations also in the germline of the developing embryo. In the next few slides, I will talk about potential ways to study fetal programming and identify this. So these are 4 ways that have been published so far. There are, of course, many more, but I will highlight on these 4 for now. I will start with Famine cohorts, and Famine cohorts are absolutely very informative to provide information about how fetal programming can impact the future health of the future generations. And one of the most extensively studied examples is the Dutch winter hunger famine. And this occurred between 1944 and 1945, and this happened towards the end of World War II, where food supplies became very scarce in the Netherlands. And then the winter set in, the canals froze over, and it was impossible for barges to function. The railway lines were shut off, which means that there was no food coming in. There was immense shortage of food. And offspring of these people who were exposed when they were born after the famine were actually much smaller than the regular babies in the Netherlands. And there was a high risk of low birth weight in babies. And this persisted for 2 generations subsequently. What was also seen is that the prenatal famine exposed individuals had higher BMI, higher serum triglycerides, higher fasting glucose as well in later life. And recent studies, and there are many, many studies about this, and one such study is this one, which has actually identified the epigenetic changes that have happened. So these are each of these, so this indicates the significance level, and the more higher the dots are, the more significant the values are. And each of these indicates 1 CpG site and methylation at 1 CpG site. And you can actually see that from the p-values that there are a number of CpG sites whose methylation states are altered, and this mediates the BMI in these offspring. And this is the same interaction for the triglycerides. It has to be remembered, of course, that there is a U-shaped relationship between birth weight and risk of type 2 diabetes. So if babies are born at very low birth weight or very high birth weight, both of these increase the risk of diabetes substantially in later life. So another way to study fetal programming and its effect is through birth cohorts. And I here present 2 very elegant studies. The first one, which of course has to be mentioned, is the PMNS cohort. And this is the Pune Maternal Nutrition Study, which was conceptualized and by Professor Chitranjan Yachnik and initiated in 1993, so really early on. The participants here were recruited and followed for 2 generations now, and soon it will be 30 years since the conception of this study. A number of important discoveries have been reported through the study, and one of the seminal findings is of course the description of the thin fat phenotype, wherein the overall weight of an individual or the baby was very low and the muscle mass was low, but there was very high fat mass compared to the European counterparts. A large proportion of the young men and women also showed abnormal glucose tolerance, and they also the low birth weight individuals had higher glucose concentrations. So another thing that came out of the study was the impact of the maternal micronutrition status and physical activity during pregnancy were also related to the child's anthropometry and had a significant effect. And I have a very wonderful collaboration ongoing with Professor Yachnik and his team, and I'm delighted to have the opportunity to continue working on this cohort to look into some very interesting scientific questions related to fetal programming and parent of origin effects. So a second study that I have been part of as well is the Poner Study, as it's in Swahili, and this is based in Tanzania. So this involves collaborators from Tanzania, Denmark, and Sweden. And this study is based in Koragwe, Tanzania, and there's a high prevalence of anemia and malaria in this population. And what's been observed is that this is sort of accompanied by high rates of cardiovascular diseases and cardiometabolic disorders in later life in this population in subsequent generations. So we here studied fetal programming in the offspring due to maternal anemia, malaria, and GDM and other exposures. And I will briefly summarize what has been found so far. So it has been found that anemia and malaria during pregnancy impact fetal-placental vascularization. There is also a high prevalence of gestational diabetes in this population. And anemia and GDM, of course, in older generations, concordant to what has been reported before, has also been accompanied by higher cardiovascular diseases in the younger generations. And what we have now done is we've performed a lot of omic studies in the cord blood and the placenta of these individuals. Well, placenta is next, but in the cord blood, and we find a lot of signatures of fetal programming in the cord blood. So there are gene expression signatures that are accompanied by epigenetic signatures or DNA methylation changes because of these maternal exposures in the cord blood. And these relate to how these are important developmental genes that modulate the development of specific organs like the pancreas or beta cell development. So there are some exciting findings, and we've submitted this paper and hope to have it accepted soon. So other ways to study fetal programming include animal models and family cohorts. And these are two things that you must have, of course, if you would like to look into this. And I will show you some data from these two methodologies combined. So I'll start with this one. So this is KCQ1. This gene encodes a protein for a voltage and lipid-gated potassium channel. And these voltage-gated potassium channels have been shown to have diverse functions, including maintaining membrane potential and regulating cell volume, modulating electrical excitability in neurons, etc. So multiple functions have been reported. And genetic variants in this or SNPs in this gene have been shown to associate with type 2 diabetes risk, primarily started with the Asian population, but replicated robustly in the European studies as well. And then this really cool study was published about a decade ago, which originated from Decode Genetics in Iceland, where genetic variants in this gene were shown to have parent of origin effects. So what this means is that if you have pedigrees, and this includes, let's say this is a trio, right? So this is a father, a mother, and a diabetic child. And what you would expect is that in Mendelian transmission, you would expect 50% of alleles from the father and 50% from the mother. But when you actually when you see parent of origin effects, there's a significant deviation in these transmissions in that if there's a maternal effect, the transmission from the father is much less than, significantly or statistically significantly less than 50%, whereas the transmissions from the mother are significantly greater than 50%. And what was found for these variants was that the type 2 diabetes risk alleles came more often from the mother than from the father. And when they looked at gene expression patterns, it seemed like the paternal alleles were actually imprinted and maternal alleles were expressed. So this study was one of the first studies that we also tried to replicate, and we have access to the Botnia Cohort, which is from my postdoctoral mentor, Leif Group, who's the PI of this study. And this Botnia Cohort is based in Western Finland and Southern Sweden. And we had access to approximately more than 2,000 families from the Botnia study, including 4,200 individuals. And we tried to replicate these findings from the Iceland study and found that the risk allele C seems to be transmitted more often from the mother, and it replicates this data perfectly. Whereas the transmission from the father doesn't seem to differ so much between the two alleles. And this difference between the transmissions from the father and the mother are statistically significant. So we were able to robustly replicate the findings. Moreover, what we also found was that the maternal C allele, and this is studied now in non-diabetic individuals because treatment can alter the insulin values, or the OGTT values. So what we did was we took the non-diabetic children of these families, and we tried to study how these associations are occurring. And what we find is that the risk allele C, for example, from the same SNP actually increased to our glucose and fasting glucose and decreased to our insulin when it came from the mother in the studies during an OGTT. And then there were a few follow-up studies on this. So one of the follow-up studies came in from the Oxford group led by Anna Gloin. And what they found was that there was a lot of epigenetic changes that accompanied this genetic transmission. And there was a change in the programming. So in the fetal pancreas, only one copy of the allele was expressed, whereas in the adult, the expression was biallelic. So both copies were expressed, which means that there was a change in the programming from the fetal to the adult stages. And it was sort of inferred from that this risk of type 2 diabetes was mediated pretty much in early development for this gene. And then this really interesting study that was published about 5 years ago in PNAS, where they took animal models to demonstrate this. So when they took control mice and they took a knockout mice of the KCQ1, so homozygous or heterozygous knockouts, there was absolutely no difference on beta cell mass or function. So there was no difference in the size of the beta cell mass. There was no difference in the number of beta cells, and there was no difference in insulin secretion. But when they took control mice and they divided the knockout mice into paternal heterozygous knockout or maternal heterozygous knockout, then the beta cell mass was significantly decreased at birth. And when they followed it up after 24 weeks, this still persisted. So there was a reduced beta cell mass only in the paternal heterozygous knockout, but not in the control in the maternal. And this is some more data. This is actually the data from the same study, where there was an allele-specific mutation or truncation in the KCNQ1. And you can see from A that this showed a reduction in the KCNQ1 levels. And B and C also show how there is a reduction in the beta cell mass, a significant lowering of the beta cell mass only in the paternal heterozygous knockouts, but not in the wild type or the TM, which is the maternal. And this also was accompanied by changes in the expression levels of the CDKN1C, which is another neighboring gene, which is very important and sort of works in tandem with KCNQ1. And when the OGTT was performed on these mice, there was also the mice with the paternal mutation had a significantly lower glucose levels and significantly higher glucose levels and significantly lower insulin levels. This reduction was not seen in the knockdown or the knockouts in the maternal or the non-specific or the control groups. So I would conclude by saying that identifying these molecular signatures of fetal programming will provide invaluable insights into the mechanism by which the risk to disease is altered. These can also provide to be invaluable biomarkers to understand what exactly is dysregulated in the system. And early identification can provide a window of opportunity for intervention to preserve health of not only the mother and the child, but also for the subsequent future generations. And future generations might find our current way of life to be reflected in their genes. So I would end with thanking the team, the collaborators, the study participants, the funding agencies, and thank you very much for your attention. And I'm happy to take any questions.